Hey, we're going to go into these steps of DNA replication in a little more detail in this series here. Okay, so the first step, this is easy. Helicase comes in and he unwinds the DNA. Then we have, and you got to know all these main characters now that you're in Big Boys and Girls and in AP Bio. You need to know, then we have these single-stranded binding proteins that keep it open. Okay, they hold this DNA strand apart while they're being replicated. Okay, the second step is we have primase comes in. Now, remember I told you DNA polymerase is a prima donna. You have to have this RNA primer come in and lay down a little bit of primer on this because it's an actual RNA sequence, because DNA can't be added without a little bit of RNA added first. Okay, this is an evidence that maybe RNA was the original heredity molecule, because DNA won't do anything unless it's got a little bit of RNA in front of it. Okay, so primase has to start every sequence, and it adds, it's called RNA primase, and it adds a little RNA primer right here, if you see. Okay, the third step is it's like DNA polymerase is like, oh, lovely day, now I can show up because the stage has been prepared for me. So in comes DNA polymerase, and your book talks about DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. I don't have you distinguish that on your test, who does what, but it is DNA polymerase 3, and they're different, that actually adds the new bases on, okay? So here we are adding the bases, and if you look, we're adding it to the 3' end of the molecule. Watch as this guy comes in. There he goes, one more. Adding to the 3' end, so this must be the leading strand, right? This would be the parent, this would be the daughter coming in, okay? So, leading and lagging strands, who is this guy? Who's the one that separates it? You should know by now. That's going to be helicase, this yellow guy. These little black circles are those are our single-stranded binding proteins, right? That keep it open. This is the replication fork, okay? And it's always going in this direction. Okay, so of these two strands, who would be the leading and who would be the lagging? We know that DNA polymerase can only add to the 3' end. So if you look, you're going to want, and these two strands have to be anti-parallel. So down here, this guy is going to be our leading strand. Why? Because the 3' end needs to end up over here, right? So it starts with the 5' on this side, and we're adding to the 3'. Everyone see that? Okay, so this guy just gets to flow straight through, and DNA polymerase zips on through. Okay, after that RNA, and it only needs one RNA primer to be added on. Now here's our lagging strand. Who's in front? The 5' end. That proposes a problem, right? Doesn't work. So we've got to come up with something different. We end up going in the opposite direction as the replication fork. Everyone see that? The replication fork's going this way. So this is where we pull extra out, and we're going in the opposite direction, and we're adding to the 3' in fragments. Here we go. Here we go. So we would say the lagging strand adds the bases opposite the direction of the replication fork. Okay, so there we're adding to the 3' end. We have these fragments called Okazaki. Okay, so this is our limit of DNA polymerase. It can only build to the 3'. So that's why we have these Okazaki fragments. Here's a picture of Dr. Okazaki. Just kind of fun to see him. Now, what do we do with these fragments? Because that's not going to work out. Well, DNA ligase comes in, and it basically sutures these fragments together, okay? So here we would call the leading strand, and it's continuous. Up here is the lagging strand. And again, it's joined together by ligase. It's like a little spot welder, comes in and fixes the little holes in between. All right, so what do we do? Here's our ligase here. Now what happens, because remember, DNA polymerase can only work if it's adding to RNA, so then we've got to have somebody replace these RNA primers. And with the Okazaki fragments, there's a lot of RNA primers on there because you have a bunch of segments. So that's where we have DNA polymerase 1 come in, and he gets rid of the RNA primers. Okay? And he gets rid of the RNA primer on the 5' end as well. On the leading end as well, right? So, DNA polymerase removes these RNA primers, and then they replace it with DNA nucleotides. And then the strands are glued together by ligase, okay? Now, this is a new concept for you guys. This idea of chromosome erosion. It's so cool. But a lot of you all will ask, well, okay, if you use one of the old and one of the new, how old is some of the DNA in my body? It can be very old. However, your body realizes that there's a problem with that. You don't want DNA that's too old in there. So it's got like a ticking time bomb that eventually a segment of DNA will say, okay, this is where we have self-destruction. This DNA is too old. And what kind of records how old DNA is this chromosome erosion. So every time that this RNA primer is removed, which we can do with the lagging strand in the middle, right? But there's a problem. The RNA primer cannot be replaced at this end because, see, over here, we can replace this RNA primer because there's a 3' end over here, right? But there's a problem right here. There is no 3' end to add the RNA primer to. On the leading and the lagging strand, if you look here, here's the lagging. There's no RNA primer in the front. So what's going to happen, or there's no 3' end on the front to add, we lose a little bit of DNA with every DNA replication, okay? Because again, it can't even, DNA polymerase cannot add to that 3' end. So the bases at the 5' ends are lost in every replication. The long and short is, the chromosomes get shorter with each replication, and that is going to limit the number of cell divisions. But don't worry, our body's planned for that, and we have these things called telomeres. And these are just a bunch of repeating non-coding sequences at the end of the chromosome. They're a cap, right? And once the telomere, so these can get chopped off every time with no consequence to the genes that are coded for because they're non-coding. But there is a limit. Once these telomerases reach the end, well, that limits how many divisions that DNA can go through. Okay? Now, there is an enzyme that can extend the telomeres. It's called telomerase. Okay, so you have the telomeres, and then there's this enzyme telomerase that will extend it, and it adds DNA to the 5' ends. Where does that happen? Not very often. Stem cells, because that's where they talk about the growth factors that stem cells have with them. Well, what stem cells have with them are these telomerases, this enzyme that allows cells to add a little more cell division on these telomeres, okay? It extends the telomeres. Now, when can this go wrong? Cancer cells. Cancer cells have been shown to have a high number of this enzyme telomerase, which means that the telomeres are always being extended and we have unlimited divisions with cancer cells. That's a big problem, okay? That's one of the characteristics that allows cancer cells to defy odds and keep dividing. That's why Henrietta Lacks cells are still alive long after she's been dead. These two videos are going to come up when you open up your Google slides. I would suggest looking at them. If the links don't work, don't worry. Just skip through it. All right. RNA, there is a test question that asks RNA, what is our hypothesis that RNA evolved before DNA? Well, the answer to this is that RNA polymerase uses a primer, or DNA polymerase, uses a primer made up of RNA. And because DNA can't just start by itself, we have to have this RNA primer. We think that maybe RNA can replicate without needing a primer that is made up of something else. So we think this idea that you need DNA polymerase has to use an RNA primer is another example of why we think that RNA did evolve before DNA. Okay, I'm going to stop here. Hopefully you can complete your worksheet on DNA replication. Good luck.